Before you guys start, I just want to, first of all, thank the student organizer for organizing this workshop and all the activities that you are going to have available to you over these 4 or 5 days. I lost track of the number of days of the meeting. But also I want to reiterate, if you were at the pizza party last night, I mentioned something that is extremely important. The future of the society, it's you. So we really need you to tell us what you would like the society to be. So I already mentioned that there is a survey going on. If you attend the sessions at the beginning of each session, if you come 5 minutes before the beginning, there are slides that they are rolling slides, and there is a QR code for a survey. So first take that survey. And then I would like to stress the importance of activities like this. So the society really builds this activity for your growth, both professionally and personally. This is a professional opportunity. You have experts in the fields that they're going to share with you best practices and how to best proceed in this specific field of knowledge. So take advantage of this opportunity. And if you were attending the keynote speaker and the award ceremony this afternoon, both Lifetime Achievement awardee reminded us how really you had the opportunity in this society to interact and to get to know people in the field that they are expert, that they have plenty of experience, and they can share that experience and knowledge with you. So take advantage of this opportunity, enjoy the meeting, enjoy this workshop, and that's pretty much all what I have for you. Thank you.
So thank you all again for coming. And if you weren't here before, if you don't have the code and the instructions already open, use the QR code now to make sure you have them up on your laptop, because you're going to want those open. And then also, I'd like to just give a quick introduction to our first 2 speakers tonight. David Briner, who is the Senior Manager of Business Development and Planning at the McDonald Genome Institute, and Michael Hines, who is the Director of Research Operations in the Genome Technology Access Center at the McDonald Genome Institute as well. So thank you guys for coming and sharing your knowledge with us.
Not familiar with this one. How do I move the slides? Oh. I can't do that, Dave. How we all doing? So my name's Michael Hines. I've been at Washington University now for about 22 years. Working at the McDonald Genome Institute. Been doing a lot of genomics during that time. Our facility is pretty large. We are the what's that? I'm not going to start talking numbers, Dave. So our facility is the derivative of what was originally called the Genome Sequencing Center, which was part of the Human Genome Project. I think you guys are all familiar with that. Anyways, over the years, we've evolved into not just doing big government work, but doing a lot of fee-for-service work for researchers doing all kinds of research, from medical research, from clinical trial research to basic science across so many species I couldn't even begin to mention. Granted, we're a very large medical research facility, so we do a lot of mouse and human samples, but we also do plants, animals, just all kinds of interesting things. So, am I able to advance now? Okay. Okay. So, what we're going to talk about for about 15 minutes here is just kind of an overview of what we're talking about when we say single-cell RNA-seq. Now, I mentioned the word genomics just as a general term, but you're all here because you signed up for single-cell RNA-seq workshop, so we're going to focus on that. We'll go over a little bit of a general workflow and some of the things that are kind of common in today's world as far as technology is concerned. And we'll talk about some considerations. So if you're ever going to get into doing these single-cell RNA-seq studies, there's some things to think about. And then once I've given that overview, we'll move into some fun stuff, some interactive stuff with analysis. So, what can we do with single cell that we couldn't do with other gene expression technologies? We're at a point now where we can look at this high resolution transcriptional profiling, looking at heterogeneous populations of cells. As you'll see in the next slide, I'm gonna show you, kind of the comparison, basic comparison between what we would call bulk RNA-seq and then single cell. So single cell is now probably it's the fastest growing assay that we do, to the tune of, we probably do at least 2,000 samples a year, maybe across 500 different projects, across 300 different investigators looking at all kinds of different things. And that's almost doubling every year. And we find that at other genomic centers, academic centers, and companies all across the country and all across the world. So the power of single cell is that we can look, obviously at each individual cell, and we can identify rare cell types that you can't see when you did traditional gene expression studies, whether they would be QRT-PCR, or if there's anybody in the room that knows what a gene expression microarray is. There we go. And now what's done is generally what's called bulk RNA sequencing. So the ability to uncover cell-to-cell gene expression variability, all of these things are things that we could never do before until just the last several years. At the same time, when we're doing these single-cell RNA profiling studies, we can look at cell surface markers. We can look at epigenomic markers in the form of ATAC-seq. We can be doing perturbation screens where we're actually modifying cells, looking at them in real time. We're looking at each and every single individual cell, and it's extremely powerful technology. So what is the big difference between bulk RNA-seq and single cell? Well, pretty basic. When we take a bunch of cells or a bunch of tissue and we isolate cytoplasm, when we get the RNA, the total cellular RNA out of it, we can look at one profile. And that's informative, and we have made lots of discoveries across so many disciplines over the years looking at bulk RNA-seq or microarray or QRT-PCR, all these things I talked about before. And those are powerful studies, but what we can do when we can actually tease out and look at the transcriptional profile of every single cell in a study, we can actually find these rare cell types. We can look at what we'll eventually get to when we talk about like a Tisney plot, where we can actually see how these cells are grouping based on their RNA, what's being expressed in these cells. And it's incredibly powerful and something that, again, up until recently, we've never been able to do. So how does this work? There are a lot of different methods now. And there's a lot of different companies, and there's a lot of homebrew technology that's out there too. It's starting to mature as far as technology is concerned. There's some sizable companies that do this pretty regularly. This one happens to be mentioned here. This is 10x Genomics. This is actually found, when this was discovered, it was called DropSeq. And the reason it's called DropSeq is that every cell flows into a chamber and flows into an oil droplet. And inside that oil droplet is a bead. And inside that bead, I don't know if I'm getting too technical. Somebody yell at me if I'm getting too technical. No? Okay. Inside on that bead is a series of poly-T oligonucleotides that can bind the poly-A tail of an mRNA. And so inside that bead becomes the reaction chamber. So each tube might have 10,000 cells in it that's going to get captured. And each one of those cells is in a tiny droplet, and inside that droplet is where the reaction happens. When we make cDNA, we actually barcode. Each bead has an address on it, has a specific barcode, so that when we make cDNA, we can tease out because we're going to pop all those droplets, we're going to mix them all together. It's a big emulsion of all these droplets with cells in it. And then we're going to tease that out eventually into single-cell RNA-seq data. In our lab, we read all this out on an Illumina sequencer, and then we flow into, in this particular example, like I said, 10x Genomics, we flow into an example, the software is called Cell Ranger. So we start with this tube of cells, and what we end up with is just all of this thousands and thousands of cells worth of individual data per cell. So one of the things that's probably the most important, and that's what I'm going to talk about for the next few minutes, is what kind of considerations you should make when you're doing these experiments. They're expensive, and they're often difficult, because when we think about, if you're looking at parts of a leaf or whatever, and you take that leaf and you want to extract nucleic acid and look at some profile from it, okay, that's fairly trivial in today's world. Same thing with a piece of tissue from a model organism, from an animal, say from a mouse liver. But when we go to dissociate those cells and then look at those cells individually, it's not so easy. So you have to consider what is the origin of your material? Are you working with blood? Are you working with tissues that you have to do this dissociation, or are you working with cells that are already in a suspension like blood? So, or cell culture. So there's a lot of considerations. And once you decide what your experiment is, then you're going to start to look at what type of method best fits your set of samples. In this particular case, we can be looking at fresh cells. We can be looking at fixed cells that are fixed with formaldehyde, for example, or they could be frozen tissues. And so there's a word on this slide that says timing, because timing is everything. Timing in these transcriptional studies is extremely important, because if the longer the cells are out of their normal environment, the cells are reacting differently and transcriptional profiles are changing. And so you want to consider something like that when you go to pick the technology that makes the most sense for your study. And like I said, it may make sense to actually fix cells because of something like that, because of a timing issue. The other timing that's important is if you are going to use fresh cells, those fresh cells are only viable for so long. So when we say timing, you're going to be contacting someone like me saying, when can I bring my cells by so that we can get them loaded and we can get this stuff moving? Because if I let them sit around too long, they're going to die, and that's obviously a big problem. So so think about the sample type that you have, what you're trying to do, and then you can start to pick the proper technology that you want to utilize. When we talk about single-cell RNA-seq, there's generally two methods that we'd look for. When either we capture a viable cell, or we fix that cell, or we get the nuclei and get the RNA from the nuclei, because the nuclei is basically the preliminary step before the mRNA ends up in the cytoplasm. So why would we look at nuclei? Well, nuclei can be more stable in like fresh frozen tissue. Okay, so when we go to dissociate that tissue, it may be easier to actually get viable nuclei than it is viable cells. That's one example. We run into scenarios like certain neuronal cells that are just difficult or large. They're difficult to isolate. So we might get nuclei from those kind of materials. But there's a value to doing fresh cells, and that's because the cells where in the cytoplasm where mRNA is being processed eventually into a protein, is that that there's more information typically in the cytoplasm than there is in the nucleus in terms of RNA of what's being expressed. So it depends on what you're trying to do. In some cases, you're limited to nuclei. And in other cases, it's best to just go with fresh whole cells. The other thing is cell quality. There's a bunch, like I said, there's a bunch of different technologies out there. There are these droplet-based technologies like 10x genomics. There's a technology called split-seq, where you actually fix the cell and you actually do the reaction inside every cell. It's kind of cool. The cell never pops open. You actually sort of take hostage of the cell and make it do reactions inside of itself. It's kind of neat. There's ways to sort cells and put them into plates. In all these scenarios, it's always bad if you end up with more than one cell, or if you end up with lysed cells where all the material is already spilling out, or if you have protein and all this other stuff. So you want to make sure that you're always working with as clean of a substrate as you possibly can. And what this slide is showing you is that there's a lot of different ways that you can purify cells. Probably the most commonly used that we see is what you see at the very end, where you see 4 check marks, and that's cell sorting, because that has a tendency to get rid of all the large and small debris, as well as dead cells and cell aggregates. So there are ways that you would want to consider all these different methods that makes the most sense for your sample type. And again, sample types are going to vary widely depending on what kind of research you do. If you're a plant researcher, your cells are obviously going to behave a lot better than, or a lot differently than something that comes from human tissue or something along those lines. And I don't want to go into all those details, because I think my job here is just to give you kind of an idea of what to think about. And these are what our researchers think about this all the time at WashU, and we get samples from all, like I said, regionally. When it comes to single cell, most of our researchers are from WashU because we work with fresh cells, and it's hard to you can't really ship fresh cells without them dying. So a lot of the single cell work we do just down the street here is done with our own researchers. And they think about these things all the time. They think about cell counts. They think about how does it fit the technology. And concentration and cell number are extremely, extremely important in your final result. You don't want to have too little. You also don't want to have too much. So cell counting becomes an important thing. I mentioned the cell sorter in the last slides. Almost every cell sorter will give you an estimate of how many cells you have in your final tube. But it usually overestimates that cell number. So should you do additional counting? Sometimes the answer is yes, sometimes it's no. You might already know how the cell sorter works on a particular marker when you've sorted a particular set of cells, and you already know that that cell count is good enough, but sometimes you don't. So if we want to talk about manual methods like a hemocytometer, that's great. But you need to do it multiple times in order to get accuracy, because doing cell counts is not a trivial thing. It's not always reproducible. You need to do multiple counts. The best thing you can do is fluorescence, which is what we do. Great, right? Why doesn't everyone use fluorescence? Well, you have to buy like a K2 ceilometer that you see listed on the slide. Well, that's not a cheap instrument, and then you have to pay staff like ours in order to do it. So timing, sample type, money, all this stuff factors into how you're going to set up your single-cell RNA-seq experiment. And so here we get to the end where we're talking about practical considerations, which comes back to money. In the case of 10x Genomics, a lot of the we load about 20,000 cells into their legacy gem, as it's called, and we might capture, let's just say, 50% of those. So that's about 10,000 cells, and then we're going to target about 50,000 reads per every cell. Well, that translates into money. And between the capture, the library prep, the sequencing, and everything, it might be $3,000 a sample. That one sample is 10,000 cells. Well, typically you're looking at multiple samples. So you can see how this adds up pretty quickly. So all of your preliminary work and your planning is critical for success. And so the way that we do this is that there are researchers that they know all this stuff. They've done it before. They're just pros at it. They don't talk to us, and that's fine because they're usually successful. Junior researchers that have never done this before come to us all the time, and we encourage them to do that because we want them to be successful. As a core facility, we're not in charge of their samples. We're just trying to give them the best data that we possibly can. So we encourage them to contact us, talk about all these problems or these potential issues that could arise. And you'll find that almost any company or any academic core facility or anybody that does these kind of things, they will encourage these conversations up front because they're critical for success. The second thing is, the same thing holds true with the vendors. They want to sell as many reagents as they possibly can. They want their name on that publication, right? So talk to them. They have a lot of amazing resources on these sites, especially when it comes to tissue dissociation. And like I said, there's a lot of people in this room that have probably, we're not just talking about human clinical trial samples. We're talking about all kinds of different things that you might end up studying. And so there's only, the power of your success really relies on what people have already done and figured out. And these companies are a great resource for that. All of this stuff needs to be considered for your experimental goals and your prep methods and things along those lines. And when you consider, when you look at the big picture, when you ask questions and do the research up front, you're much more likely to be successful into generating meaningful RNA-seq data. So with that, that was kind of the overview I wanted to give. We've got some backup slides that we can go through if we want to talk about more of this stuff, but maybe I'll just stop right there and just say, does anybody have any questions?
Thank you so much. We appreciate the talk. My question is about the barcode. Mm-hmm. I know the Illumina, they use usually double barcode for increase the number. So when you're saying about like 700 cells or more, how this barcode's gonna work for be really unique like?
Okay, so what's typically done is this. So when I said barcoded cells, that was kind of a simplistic version of what actually happens. It's pretty complex the way these libraries are made. So if you know Illumina sequencing, and this is the way we always do it, we do what we call a paired end read. So every library fragment gets read from one end and then from the other end. And so the way that in the slide I showed, I guess I can back up. I don't know if it's gonna really, yeah, this isn't very descriptive at all. When we talk about barcoded cDNA, we're actually, during reverse transcription, we're going to incorporate an oligo, and forgive me here, I think it's at the 5' end. And so that ends up on the end of the library fragment. When we go to make the actual library, all of the barcoded cDNAs are pooled together. And just like any other Illumina sequencing, or actually the newer sequencing companies work the same way. I don't know if anybody's familiar with Element or Singular. They're all, you ligate a sequence adapter onto the end, and that sequence adapter contains, in the case of Illumina, it contains 2 unique dual indexes. And that's just to decipher what the library is. So when we do multiple samples, that might be 10,000 samples, each sample has a set of unique dual indexes, but each cell has its own barcode that's read out in read 2. There's read 1 and read 2. Read 1 reads the transcript, read 2, I think I got that right. Read 2 reads the barcode. And then the indexing, so there's actually 4 reads on every library in an Illumina experiment. There's 2 index reads that tell you what library it belongs to, and then there's read 1, which tells you what the transcript is, where it aligns and what it is, and then read 2 tells you where, what cell that belongs to. And all this gets deciphered bioinformatically, and I don't know if that's covered in today's workshop, but that's a great question. It's complex, and not all of the technologies work exactly in the way I just described that. I hope I did okay with that. But yeah, you always have to find out what library it belongs to, and find out what cell it belongs to, and then there's always a read to figure out what transcript you're actually looking at. Yes.
Thank you. I mean, your barcoding, if I understand correctly, is to have homogeneous material for doing sequencing. But how you call it then your droplets? My question would be, what is your minimum amount of RNA material? I mean, I understand it should not be cells exclusively. How much minimum RNA material do you need in your individual droplets to do your sequencing?
It's literally, so when that droplet forms with the bead and the cell lyses. So whatever is available in that cell is what you have. That's the limiting, that's the rate limiting
how much
step.
RNA material do you need minimum? RNA material, because this is what you sequence.
Right, but we're not extracting RNA. We're actually doing the experiment.
I know. I mean,
So,
you
yeah,
can
I'm
work
sorry, I'm
with
misunderstanding
nuclei,
your
you
question.
can work with cell lysate, whatever it is, but what you finally will sequence is RNA material. So how much minimum material of RNA do you need in your individual droplets?
The minimum cells to go in, it depends on the technology. In this particular example, we would load, it's about 10,000 cells that would go into this experiment.
Yeah, but 10,000 cells, I mean, if I give you a cell population of 10,000 cells and there are 10 different cell types, means an average of 1,000 cells per cell type.
Okay?
I expect this is not enough for doing your sequencing.
It is. It is. It very much is. It is. Trust me.
Of
We've been
course
doing
I
these for
trust
years.
you.
Yeah. So think about it this way. And it varies per cell type, and I don't want to get into, some cells might only have a picogram of cytoplasmic RNA. Others will have 100 picograms or whatever. This technology is literally, and you'll see that in the data. So the other thing that I didn't mention is that there's actually a unique molecular index also incorporated into the library that tells you that you're not reading the same library fragment over and over and over. Mm-hmm. So there's actually kind of technically 5 reads that happen with a single cell experiment like this. Mm-hmm. And what you find is that there is a limit on the number of transcripts that you can detect with a droplet-based technology. You're mostly sampling the highest expressing RNAs, RNA transcripts, as well as some moderate expressors. A lot of the low expressors you really won't pick up from this kind of technology. Now, this company just released a very new product. So you can sequence these things to saturation pretty quickly, which is why we only say 50,000 reads per cell. If you go out beyond that in terms of the read depth, you're just sequencing the same pieces of cDNA, same pieces of RNA over and over and over and over, and there's no value to doing it.
Like most things in nature, it's not what is abundant that is making the difference. It is what is in minimum amount that is making the difference. So all the abundant RNAs that are there in these different cell types, 10 different cell types, in fact, they do not interest me. I'm only interested in the ones that are in small amounts and that make a difference over my 10 cell types.
So if that was the case, if you were looking at a lot of the studies that are done with like a droplet-based technology like this are looking, they're I don't want to say they're fishing expeditions, but you're generally trying to understand the cellular makeup of your experiment using using the RNA in the cells. So you'll see this, I believe, in the analysis where you'll see these groupings of cells, and you'll say, if it's a truly heterogeneous population, and you see macrophages, and you see, I could go in, but you'll see all these different cell types come out, but you might be looking to see where there's different groupings of the same cell type, but there's something interesting there, okay? And then you might find a marker to sort on with those cells. And so then you go back and you redo an experiment, and you actually use a cell sorter, and you're going, okay, I want to look and see more specifically what's going on with that subset of cells. And you put it on a sorter, and then you want to sequence it again. You may not use a technology like this. What you may use, a cell sorter can actually deposit a single cell into a well of like a 384 well plate. And then you can do another technology called SmartSeq. Now, SmartSeq actually does sample most of what's in that cytoplasm. It's an amazing technology that allows you to amplify at a single cell level the contents of what's in that cell. So a technology like this, think mouse and human, an average cell might express let's just let's just say 15,000 different transcripts, or sorry, genes. 10x genomics, like this droplet-based technology, it might only sample 5, 6, 7,000 of those cells, because stochastic, there's something with those low expressors, we just don't pick them up. But if we do a different technology like the one I mentioned called SmartSeq 3, I think we're on SmartSeq 4 now, where we deposit these cells and then we actually do a reaction on every single cell. And we can actually look at full-length transcripts on those kind of things too. We can even use long-read sequencing. There's a lot of different things you can do when we get into different technologies. Those technologies will actually allow you to, so to answer your initial question, if you did something like SmartSeq, the answer to your question is you need 1 nanogram.
Okay, thank you. You talk about human and mice. I'm an insect person, invertebrate person. So what about your experience with, let's say, invertebrate animals or non-model plant systems?
Okay.
Out of the box, not Arabidopsis, not Drosophila, not Chinorhabditis, just something that is a little bit more interesting.
Yeah. We don't do a lot of that. We do some. And in cases where there's not a good reference alignment, so if there's not a good reference genome and an annotated transcriptome to go along with that reference, it's going to be pretty difficult. If you do a short read technology like this, you're mostly just doing transcript counting. You can't do anything. You can't do de novo assembly. You can't do any of those sort of things. If you do, again, going back to something like SmartSeq, where you could actually look at long reads. So so that's really the first question. Do you have a reliable reference genome, and is it annotated well? And if that's the case, as long as it's got a poly A tail for a technology like you see on the screen here, it'll work.
Yeah, of course. That was my last question.
Okay.
We'll have others, but
It's a good question, just how sensitive can you get to one cell?
Yeah.
How much of that messenger RNA can you measure, right? So those are good questions. Rand Snout may be zooming in later. He may be able to help me more with that. It's an evolving technology,
Yeah.
and there's no one approach, as you mentioned.
There's a bunch, yeah. There's a whole bunch.
We have a lot more to this workshop.
Okay. All right.
Next.
All right. Thank you all.
Thank you.
Thank you for sharing the introduction with us, and that concludes the introduction portion of the workshop. And now we're going to go on to the hands-on portion, and we have Rand Snout on Zoom to run it through with you. And as you guys have questions as we go through this, just raise your hand, and we'll be walking around with the microphone so that you can ask questions and things throughout all of it. I also want to make a point that this is just to give you a general understanding of single-cell RNA-seq. There's a lot to it. You will have all of these resources available to you. So after this, you can continue to go through it. We also have videos that walk through each and every step, so you can go through it more in depth to help you understand it more and more after this session as well. So thank you, and we're going to start the next part now.
So yeah, thanks for the introduction. We'll go to a hands-on sort of walk through the analysis that would go in a typical single-cell data set. As was sort of mentioned earlier as well, is that there's a lot of technologies, a lot of different ways to do this. And so you always kind of want to think about what you're actually using and using those specific workflows. But you should have seen those two QR codes, and hopefully you had time beforehand to download the sample files. So what I will do is I'll share my screen, and I've prepared, so I wasn't sure if I'd be able to join you. I wish I could be there in person, but I've got some videos, and all of these code and everything and files, they're all available to you after the workshop as well. You can walk through them again, rerun them if you run into any issues today. And then I think there's people in the room that are going to walk around as well and try to help. And so we'll go right to it. I'll share my screen. Okay, so we'll play some. I'll play the videos, and then after that video plays and we walk through with it, I'll open it up for a few questions. And so you should be able to hear this.
Hello, everyone. Welcome to this hands-on workshop on single-cell RNA sequencing data analysis and visualization. My name is Ryan Snow, and I'll be walking you through the key steps of this tutorial. Unfortunately, I couldn't be there with you in person today, but I hope that the videos and the documentation that's included in these walkthroughs, these tutorials, can be a valuable resource to you and explain more or less what you're trying to do. The goal is that by the end of this session, you'll be able to process raw sequencing data from your own single-cell experiments, if you did your own, or from someone else's single-cell experiment that you can download from any public repository, and then get to the point of creating some initial UMAP visualizations for clustering and cell annotation, and potentially even looking for cell type markers and differential expression. So we're going to kick things off with just preparing for this workshop. And so you can find information on how to get started at this GitHub link, which hopefully you would have received along with a Google Drive link, where all the data files are. So this analysis requires quite a few software installations and files. And so we'll be using Cell Ranger Pipeline, which is one of the most commonly used platforms today. This walks you through how to get those contents and how to get those packages so we can actually get at least most of it right. So the very first step that you want to do is copy the contents of the SIVB shared Google Drive. You should have received the link via email. And this can take up to 1 or 2 hours to copy because there's some pretty big files. These are example data sets and things that will allow us to move forward, whether there are hurdles along the way or issues along the way. So to do this, what you want to do is download the folder of files on the Google Drive. That's a shared drive. And you want to download that as a zip folder. It should come up with a zip folder, and you want to extract that on your using a typical method on your computer operating system. Usually it's a right-click, extract all. And inside of that folder, you should have something called SIVB workshop files. You then want to log into your own Google Drive, and here's a really important note of caution here is that some institutions may have some limits on what their Google linked accounts can do. And so you might actually need for this workshop to have your own personal Google account or create your own just for the purpose of this workshop. And the limited space that you have on a create account should be enough to get these files, but you might also run into some space issues. So these are things to be cautious about. You want to click on My Drive, New, and Upload folder. And when you upload that folder, you want to make sure that you upload the folder called SIVB workshop files inside of that extracted folder that you downloaded. So that means that after this, you should have a folder called SIVB workshop files, and it should not be nested within any other folder. So if you click on My Drive, it should show up right there. And it will take about 12 gigabytes of space. Next, you want to download and upload the Google Colab workbook, which I'll show you really briefly how to do that. Essentially is through this GitHub link, you want to download the file called SIVB Workshop 2024 IPYND. Download that file, and then go to Google Colab onto your Google Colab account with the account you just made or where you uploaded those files, and upload this notebook. And all you have to do is go to File, Upload Notebook. And then you want to test the drive connection, which I'll show you briefly how to do that as well, where it will ask you for permission to be able to access your Google Shared Drive. You want to say OK. And then after that runs, you should see this output right here. So if all steps completed successfully, you should be able to run this tutorial in this workshop or even at home. And again, the note that I want to, because we want to have everyone kind of work on the same environment, computational environment for this workshop, we're using Google Colab, which is a free cloud service. But as expected, there's some limitations to this. And so keep this in mind as we go through it. There will be some parts that will crash or not let you not finish. And I put notes as much as possible where this happens, but you should still be able to run through everything and get a general idea of how to run this. So let's go to download this file. So you can click on this SIVB link right here. You can download on this IPYNV file right here. And then on the side here, you should have an option to download the raw file right there. And that will download this for you. Okay, so here I've already uploaded that IPYNV workshop file. You can see this right here. And this is what this workshop file looks like. It's a hands-on single-cell RNA-seq data analysis and visualization workshop. This YouTube video is right at the start, and you can read through it. And again, this is intended to be a simple hands-on walkthrough for the analysis of single-cell RNA-seq data. And we essentially explore three main steps, which is setting up your analysis environment, alignment of sequencing reads, which is section 2, and preliminary analysis in Python, which is section 3. So as I mentioned, you want to be able to run a specific chunk of code to make sure that everything works. And this is the block right here. So I will clear my output so you can see this working. And you hit this run button on the side here. And this should run. And you'll see that I already mounted this, but it already asked me for the permission to connect this. And it shows the files that I have in my directory. Those were the files in the uploaded folder. And if all of this works, then you should be able to run through the rest of this. So thanks for following along with this step. We're going to have some more videos along the way. This step really was to kind of get everything set up and make sure that we can actually get these files uploaded and connected with the workshop Python files so that you can actually run this. So I hope you enjoy the rest of this tutorial. Thank you.
Okay, so let me share. We'll go to the next step here and let me share. Is it full screen?
Yes.
Okay. Segment, setting up your analysis environment. Okay, so welcome to the next segment, setting up your analysis environment. In this segment, we'll dive into the second key step of our single-cell RNA sequencing data analysis workshop, which is the alignment of sequencing reads. So by the end of this section, we hope that you understand how to align your sequencing reads and interpret the output of the Cell Ranger tool that's used to do this analysis. As I mentioned, this workshop, we'll use a 10x Genomics Cell Ranger software because it's probably one of the most widely used platforms right now, and there's a lot of sample data sets that we can work with. But you can find very specific tutorials that walk you through very much some of the same steps that we're going through that are specific to certain vendors. So 10x Genomics has some, Parse Biosciences, Curio Biosciences, which are just three of the many single-cell platforms that are out there. So I'm not picking or choosing favorites. It's just three that are pretty common. And you can click on these links in the notebook, and it'll take you to those websites and some idea of how it runs. Now, some of the useful tips as we go through this analysis code is that in this interactive notebook, you might see character exclamation points used at the start of several lines of code. The reason we do this is because we don't have direct access to the command line using Google Colab. Because this is a free account, we don't have that access. It's not our own Linux server, so we're quite limited, as I mentioned before as well. But these are some of the lines that you could run at your own institution if you're given access or you somehow get access to a Linux terminal, or through like Microsoft Azure, AWS, or any other type of cloud provider that allows you command line access. Because some of these things you do need to run on the command line and not in Python. Now, we are able to trick this notebook into running these command line code using that exclamation point. The other thing I want you to keep an eye out is for these indented notes that look like this useful tip here with the gray. This is where we'll indicate steps that might take a little bit longer than 10 minutes to run, or if something's expected to fail on this free account. Don't worry if something fails. You can keep going. The next steps will work. One of the reasons we copied the files over is that we can keep just kind of skipping the steps. We're saving the up, these steps so we can keep going. Okay, so let's dive right into it. So as I mentioned, we'll be using Cell Ranger. And so the first thing we want to do is download and test Cell Ranger. And so the following code describes how to install that. And we get this link for downloading it from this 10x Genomics server. It'll ask you to log in. So for that reason, we don't have everyone downloaded directly from the 10x Genomics server. But what we do is we can run this wget command on the command line with a link to the Cell Ranger, which is an HTTP link. And then we have to extract that. So to do that, what we did is we actually copied this installation file right here. So you can see that's the exact same thing. Well, somewhat the same thing. We got Cell Ranger 8.0. And then we extract or unpack that Cell Ranger. And then the first thing we want to do is actually run a site check. And that site check just makes sure that the compute that you have access to has the right resources. Now, when we run it here, it'll tell us that we don't have the right resources. That's not surprising because it's a free account. But this is something to have access to whenever you run your own analysis so that you know that when you run into problems, if you do, that it wasn't because of some sort of problems with the compute resources you have access to. So let's go ahead and run this and have that file copy over. It'll extract or unpack. And then we'll just print out the simple help just to make sure that it actually can run. So if the help runs, then we should see a certain amount of output here, which we'll get to in a few seconds. Again, this could take some time or not, depending on access to the resources that we have. Great, so we can see that a help output came out for the 10x Cell Ranger software. It tells you things that you can do, like you could run this mkfastq, you can run multi-count, just different ways of using this command. So we know that it copied and unpacked correctly. So then we can run the site check. And this ran pretty fast. This site check.txt file is saved, so we don't actually look at it, but you can access your files on the side here, and you can see that there's a site check.txt. And you can download this file and take a look at it if you ever wanted to. Next, what we want to do is do a test run. So we'll run the following code, and it should state that the pipe stance completed successfully. And that means Cell Ranger works as expected. Now, this can take 20 minutes or even more, and sometimes might even fail on a free Google Colab instance. It shouldn't fail, but it should take about 20 minutes. So I won't run this right now because I don't need to. But you'll want to run this and just sort of take a look at this check install.txt output. And it should tell you that it's a completed successful. So now we get to the actual analysis aspect of it. So we've installed the software. Now, the plan here is to actually align the raw reads that you received from whatever sequencing service that you have or wherever you got that data from. Maybe you downloaded it from a website. And we want to align that to a reference genome to be able to tell what RNA was detected. And so to do this, we need the reference genome. So most of the times, people will use human, mouse, rat, Drosophila. And when that's the case, those common model organisms, vendors have previously built custom references, which can be downloaded directly. In other cases, we might need to build our custom references. And so because some of you might be working with sort of non-standard species, what I just want to go through really quickly is how would you build this for a non-reference or a non-model organism? And so here in this example, we copy and unpack. This is a mouse, GRC-M39. And so we'll copy that as if we downloaded it. So this would be the link right here that we would download it from. And then we're going to unpack those files as well. So this is an example for a model organism. And you can see that it's pretty simple. It's already set up as you need it. And again, it depends on which species you use. And this shouldn't take too long. Again, depends on the size of the Okay, so we saw that it started. I stopped it because it is taking some time here. We'll wait about 2 minutes here. You can wait. Again, as I walk through this, I might just skip some steps. And again, if some of these fail, it's not a big problem. It just gives you an idea, right? So what we did is we downloaded the mouse reference genome and we unpacked it so we can use it for the analysis. Now, what about a custom reference genome? So here, for example, we'll use cow. So we can get reference data from the Ensemble FTP server. So you'll want to download that data from a reputable source. Now, this workflow can take a long time depending on the genome size, and actually the cow will probably fail on this free instance. But what we did is we got this DNA top level FASTA file. And so we'll copy that and extract it all. And we'll get the gene models and we'll extract that as well. So this step should complete. Again, depending on the size, it might take some time. But here, what we do is we get the DNA sequences, and we also get a file, the GTF file, which says this is where the genes are, coding genes, non-coding genes. You get to pick later on. We'll see what you want to include. But this is how you sort of restrict where the sequences try to get aligned to make this a little bit more efficient as well. Great, so these copied over. It didn't really give us a message, but again, you can see on this file area on the side that we have now that mouse data. We do have those two cow data sets. And so we can see that they copied over. So next we decide, well, what types of genes we want to include when we're looking at the alignment. And so in this case, we'll only keep protein coding. When we build that reference genome using this make GTF from Cell Ranger. And so this formats it in a way that 10x Genomics Cell Ranger can use. And so we can run this as well, and this should run relatively quickly as well, but you'll see that there's a warning saying that this could take up to 10 minutes. Again, this depends on the size of the data set or of the reference genome and the GTF file. And I believe the cow is not that big, so it should finish pretty soon.
Has everybody got to where we are here? Nobody's lagging behind. I just want to make sure everybody's on the same page. Nobody needs help.
Perfect, so we're done, and that was done. So after that, oh, I forgot to mention as well, the inputs were that GTF file, and so the input was a GTF file, and the output is this new filtered GTF file. So we're making a smaller GTF file from which we want to make the reference genome. Okay, so now it's building that reference genome, and this step will most likely fail because this is a pre-analysis environment. But we run this make reference command from Cell Ranger, and we're going to call that reference genome bTau10, so post Toros 10. And the input FASTA file is this top-level DNA.fa file. And the genes is this filtered GTF file. And we're going to call this version 1 because it's the first version that we make, but you could also call it, for example, version 3 because this is our ARS UCD 1.3. Just name it something that is meaningful, that you can always make sure that you understand like where did this come from. Okay, so let's go ahead and run this. Again, you'll see a lot of this output. It's going to keep going for a while. You can see that it's starting to work here on the files on the side. As I mentioned again, this can take some time, and in this case, will most likely fail. But we will sort of let it run, and we'll come back together once this has finished or failed. And again, if you want to, you can also stop it. None of the next steps are dependent on it. But we'll get back together and talk about the alignment of the sequencing reads here after this is complete. So thank you.
Okay, so how is everyone doing? Do we need some time or go to the next video? Hopefully everything is running for everyone.
Rance, I have one question really quickly. I have a student here who's having a little bit of difficulty accessing it just between her personal or professional account. Do you mind if checking if any of the access has been blocked and you can gain access for her?
So it's to the notebook is blocked?
Yeah, just on her personal Gmail. She's accessing her the code through her professional, and she's trying to do it through her personal for the Google Drive, just because her professional doesn't have enough room for the Google Drive.
Yeah, so unfortunately that has to be done. So the same account has to be used for the collab and where the files are.
Okay, that's why we were
Uh,
hoping to gain access.
Yeah, the only way to do it is to have them together because you have to give your account access to that Google Drive. So unfortunately, that is the only way this will work.
Okay, okay, thank you.
Yeah, so hopefully you can get those files copied. It is one of the tough things about using these free cloud services, right? And so the permissions would have to be given by, the account owners, which is both, your accounts. So,
Rance, we have another question.
yeah.
Suppose if I want to align my sequences to more than one references, can I do that?
So you can do that as separate analysis.
Not in the same one.
There is probably a way to do it. I'd have to think about it because I can't think of many scenarios where you would want to do that. And so it's definitely not, it wouldn't be it's not a usual practice. So it's not developed, to do this in an easy way. So you might be able to trick the analysis software by combining the genomes together and giving it multiple GTF files that specifies, which organism it came from. So you could probably trick it to do it, but it might just be easier to do them separately. I'm having a hard time thinking of why someone would do that, but I'm sure there are some cases.
Okay, so while you're working on this, announcement that's of no use to Rance, but to you. The President Piero has invited you all up to his presidential suite, 1050. So when we're done here, you can go up. There's plenty of drinks up there, both alcoholic and non-alcoholic, okay? Sorry, Rance, no beer for you tonight from us.
That makes me wish I was there even more. Should I go ahead with the next step?
I think so, yes.
Okay.
Good.
Okay, so welcome back. In this second section here called alignment of sequencing reads, we'll actually go through how to align. So we went through the package and environment setup and cell ranger installation and building a reference genome so that we can do the alignments. So in this part right here, we'll actually do the alignments of the FASTQ files that you should have received from your sequencing port or whoever does the sequencing for you. So most sequencing cores will send you the data as FASTQ format, but in some very raw instances, you may need to convert the raw up to BCL files. And to do that, you'll have to follow vendor instructions. But usually they have a pretty well described workflow for how to do that. So let's talk briefly about what a FASTQ file is. So a FASTQ file is pretty much the output from a sequencer. And it usually consists of 4 lines with repeating patterns. So here is an example here that it just looks like pretty much gibberish, right? Nonsense. But in short, what it is that line 1 is in the instrument information. Line 2 is the raw sequence letters, so the actual sequence data. Line 3 can optionally provide you some additional sequence information. And then line 4 encodes the quality values. Okay, so we have a general idea what this looks like. And you know, when I do this type of analysis, especially when I first.
Oh, did I hear? Do I need to pause or should I keep going?
Sorry, I was.
Is everyone, I think you're good to keep going.
Yeah.
Yeah, you're good to keep
It
going.
really helps for understanding what the next steps do and also how to deal with problems if you run into them. So I'm just going to clear this up that I have from before as well. And here we do again, as we did before, we're going to copy some example FASTQ files. And actually, in this next line, instead of unpacking, what we'll do is we'll just show the first few lines of this FASTQ file, so the first 10 lines of this FASTQ file. So as you can see here, instrument information, we've got actual sequence, right? ACTG base pairs, some extra information, and then this encodes the quality information, which is a way to encode it in a way that computer can understand, you know, what it means in terms of quality. I mean, so there's a lot of software that allows you to kind of interpret what these actually mean. And you also have sort of where on the sequencing lane some of this was collected here. So you have a lot of useful information here for this data. Okay, so this is an example of a FASTQ file. So next, what we want to do is align those sequences to the reference genome that we either prepared before, such as the cow genome, reference genome, or the mouse one that we downloaded. Now, this sample data set, I downloaded directly from 10x Genomics. It's a 5,000 mouse liver nuclei. And so, of course, we're going to align this to a mouse reference genome. And so here I describe this code. So you can see right here where you can run it, that it's a long running line. So here I break it down kind of in pieces. So what we want to do is get a count. Count the number of times mRNA molecules were detected. We want to assign an ID. So we're going to call this run count 5,000 liver. We want to tell it where the FASTQ files are. And we can see in the file menu here where this is. We want to give a common string that's in the FASTQ files because usually they come in as paired ends. And we can sort of see that right here, right? R1, R2 are the two paired ends. I1, I2 are the indices. And so using this very first part of the file name, Cell Ranger knows that these are referring to the same sample. Next, we want to give the reference transcriptome, and this is the one we downloaded from 10x directly. And we can create BAM files. BAM files can be used to sort of visualize this on a genome browser. They're really big files, so here we'll say false. I put no secondary here only so that it doesn't run some of the t-SNE maps that are built in, but you can get those built-in analyses as well. And then we can provide, you know, what kind of computational resource that we want to use. So the more we can give it to run, the faster it will go. So just as a note here, this code will definitely fail because it will take hours to run. And the free cloud resources that we use don't have enough memories or CPUs. And so as part of a workshop, it's really hard to run this as an example because of just the amount of time and resources that it takes. But that being said, the code is there to run it. You could hit run and start to see the output. And you'll see that it works. It runs pre-private checks, and then it's just going to start outputting a lot of information. Or sometimes it will just fail. And that is because of resource problems. In this case, some of the files weren't in the right place, just for this specific example. But in reality, it will fail no matter what you do. there we go. So now it knows where they are. So again, there's some issues with the resources. So as part of this workshop, what I also did is I got the outputs and I put them up on here as well. So we can copy this from the Google Drive. These are called web summary HTML files. And these will actually be easier to open in your web browser. So we run this, it's pretty fast, it's copied, and we should see those files now in this folder here. I just need to refresh it. Yep, we need it to refresh. So we have this HTML file, we can download it. It's there we go, it's downloaded. And we can open this. Okay, so we have it here. So let's quickly go through kind of what we're looking at. So this HTML web summary provides a lot of useful information. And so each piece of this should be kind of examined closely with the biology in mind. And some of the key metrics you want to look at are looking at things like the estimated number of cells, the mean, median reads genes per cells, the valid barcodes, the barcode rank plot, or what's sometimes called the NEAT plot. And there's some things you kind of should expect. So let's go through this kind of quickly. So estimated number of cells, this is based on your experimental setup. How many cells did you put in and how many did you expect to have? The median genes per cell and mean reads per cell will be very tissue specific, cell type specific. I know this is a liver and actually 3,000 median genes per cell is actually pretty good. 30,000 mean reads per cell is pretty good. We can see over 90% valid barcodes, UMIs, good quality, lots of it maps to the genome. And so everything points to this having worked pretty well. Even fraction of reads in cells, 73%, suggests not a whole lot of ambient RNA. And this is actually kind of pretty well visualized here. This is a barcode rank plot. So the cells are ranked from highest number of UMIs to lowest number of UMIs. So that's unique molecular identifiers or unique RNA molecules detected in the cell. So you can see about 100,000, and then slowly, and then you have a big drop, which is why it's called a NEAT plot. And then you have almost nothing. So 110, 100 over here. And so this is how we determine what is a cell versus not a cell, because you expect cells to have a lot of different RNA molecules in them, and you expect non-cells to have almost nothing. And so this steep peak down here, this steep slope here, tells you that all of a sudden we're getting less RNA molecules in here. And that's probably where the separation between the cells and empty beads are found. We can also look at some basic gene expression analysis. So this one did do the secondary analysis, so you can already see a brief Q map, and you can see some gene expression data which you can explore, and you can also see the saturation. You want to get to the point where you're not increasing, where the slope sort of slows down, right? And this is actually looking pretty good here. So this example data shows a good example, some liver data, nuclei isolated from CD1 mouse livers. And just to kind of highlight the importance of this NEAT plot, because this is really one of the main things you want to look at. So in the following example, we have a good example and a poorer example. It's not necessarily bad, but it's much poorer. And some of the key things to look at here is that UMI counts for 100,000 here, but they're actually about 5,000 here. So we have a lot less RNA. Number of barcodes, we also detected way fewer barcodes, and so we've lost a lot of beads. Somehow, whether they weren't sequenced, not sequenced deep enough, or anything like that. Now, what this means is that you see in this data set, you can pretty clearly see where this drop or this knee happens, and you can separate the cells from the empty beads. But here it's a lot more questionable. You do seem to see kind of a knee here, but it's a lot harder to distinguish that. Now, in this case, these might be cells that just have a lot of much less RNA. And actually, I know that this tissue here that this data came from is an RNA poor sample. And so it's not necessarily a poor sample prep, but you kind of do wish that it would be a lot cleaner, a lot steeper, and like, there's a lot of factors that come into play. So these are some of the metrics that you want to look at after you align. So after running through the whole section, it's a really brief section, but really all we did is we used Cell Ranger to get the counts, to get that web summary file. And as part of that web summary file, we also got a folder with all the actual count data in it. And that is actually what we will be using in the next step, which is the Python analysis. Thank you.
Okay, so how are things going? Hopefully the analysis is running. Like I said, that one step won't run. So really it's with the web summary file that will show up. So hopefully everything's running well for everyone.
Anyone have any questions? Okay, I think we're good to move on.
Okay, perfect.
Okay, so welcome back. Here we're at step number 3, advanced analysis using SCAN-P or SCBI. I call this advanced analysis, but actually it's kind of the basic analysis in Python. But it is after the alignment, the reference, genome stuff, and all the Cell Ranger steps. So arguably, I would say this is kind of the fun part. We're getting to the part where we're actually starting to like cluster and integrate data and actually kind of get an idea of what our cells actually are. Now, just like we did for the Cell Ranger analysis and the alignment, we want to set up our environment. Python has a lot of packages that you can install. So if anyone's used it, you can have a lot of different versions, different setups. And so one thing that I highly recommend in doing these types of analysis is working with a virtual environment so that you can control and monitor packages. And I don't go deep into virtual environments here, but it's something worth looking into if you do plan to do some analyses. But one quick sort of overview of how you can do this is using this pip install virtual environment, going to your analysis project folder, creating this virtual environment with the Python version that you want, calling your virtual environment something, and then activating it depending on whether you're using a Mac, Linux, or Windows. There's different ways to do this. So it's kind of good practice to have this environment so that you can reproducibly analyze these data sets using the same package versions and everything. In this case, we're kind of working in a Python environment. Google Colab sort of set this up for us, sort of a bare bones Python environment, so we don't have everything installed. So we're going to have to install some of this stuff. But the reason we use Google Colab is that everyone should be starting with a pretty similar environment in which we can install these and kind of make sure there's no conflicts. And hopefully that was the case through this workshop, and we'll be going through this step as well. Okay, so to install Python packages, we can use pip install. Remember that this exclamation point is just a way to access the command line. So we're going to install 4 key tools: SCAN-P, SCBI tools, iGraph, and Leiden ALG. And then we're going to copy the raw outputs from Cell Ranger, which has all the count data. This could take some time. So this is a part to be patient, but you do want all of these to install. So you want to try to not interrupt this, and it should complete successfully. And so we're going to go ahead and run these and let them install, and we'll come back here once everything is installed. Okay, and we're back. So this took about 2 minutes. you can see a lot of output, but everything installed correctly, no major issues. And so now we have the files that we need. So now we can start importing those packages, right? So we installed them, but to be able to use them, we need to import. So here are links to the individual packages if you want more information about them. Scanpy, single cell, or NumPy, pandas for data analysis, manipulation, and Seaborn for visualization. Feel free to check out these links. We also set plotting defaults for 150 dpi, even though I say 300 here. And then 600 dpi for when it's exported, just so we have something higher resolution. I also import some matplot as well to allow for some plotting as well. But all of these are just used to kind of process that single cell data, but these are packages that are already developed to do this. And this should run really fast because these are all installed. Okay, so that's set up. So next we want to tell the Python where our samples are, what our samples are, and where is all the output that we want to find. So there's a lot of ways to provide the sample information to your analysis code so that it is tracked throughout the process. And here is me getting slightly on a soapbox here, but what you want to do is provide as comprehensive metadata as possible. And for reference here, I suggest that you look into the FAIR data principle for findable, accessible, interoperable, and reusable. And really the idea here is that you want to have the metadata that people can actually reuse and reanalyze, reprocess this data, and learn more from it. Because these are big projects, often a huge investment. And so we really want to maximize that return. So let's do our best to actually give people the information they need. So because we only use one sample, I'll prepare a table manually. But there's also ways where you can import tables with the following. And as an example, what will follow are the requirements from the Broad single cell program. So they have a very nice outline of at least some of the minimum amount of information that we should report as part of metadata. So we'll go with that. And so those columns are name, biosample ID, donor ID, species ontology label, disease ontology label, organ ontology label, library preparation protocol ontology label, and sex. And you see that a lot of them are matched with ontology label and species, for example. And the reason to do this is that ontologies have very specific definitions. And so to make sure that we all know what we're talking about, we can use these ontologies. And so every time someone finds this ontology, they can go to almost a dictionary with the definition of what you were talking about. So here's an example of creating this manually. So the data that we want to analyze is in this filtered feature BC matrix. Our biosample ID is L01. Our donor ID is M01. This is our species ontology. This is the ontology label, Mus musculus for mouse. That's the disease. So normal, so no disease. This is the organ, it's a liver. This is the preparation protocol. And this is the 10x 3'V3. And then the sex of the mouse. And we make a table from that. And we can actually visualize it right here. So you can see it's a table, the row, and we have all the information we just entered here. So this is all metadata that's going to be saved with the analysis moving forward. Okay, so next thing we do is we import the data and do some preliminary quality control. So the following code will do the following for each row in this table. So it's going to import the data using 10x, read 10x right here. It builds the metadata, including that metadata table. So it's going to incorporate all that information. We're going to calculate some preliminary QC metrics like mitochondrial RNA and other RNA counts and things like that. And then we're just going to collect all of that into a list. This empty list right here, we're just going to collect it to that. And we're going to create a combined data set with everything together. This should run relatively fast. You can see that we're working on L01, and if there were multiple rows in that data set, it would just keep going through each of those. Again, depending on the resources that the free instance has, it can take more or less time, but we should be finishing up relatively soon here. So you can see with this little arrow, it's still at the read 10x right here. So that would be the slowest part of the whole analysis because it's actually importing a pretty big data set. And yeah, you saw that it skipped through, went through all this build metadata, went really fast. It's calculating those QC metrics. Also should be relatively quick. And there we go. We finished this step and we have this new A data. And the way that we can check this, and we actually want to look at those QC metrics. So we'll look at some of the most common ones, which is the number of genes, number of transcripts and mitochondrial RNA in each of the cells. And so this can help us, for example, identify unhealthy cells. So those that have high mitochondrial contamination, poor quality cells that have little or no RNA, or even potential doublets that might have too much RNA. So we'll run this. We're saving that figure, but we'll also display it here. A Violin plot is almost like a histogram. Draw a line right down the middle, put it on its side, and it's a mirrored histogram. So this is the distribution along the number of genes, and you can see that there's a group of cells close to like 1,800, another group of cells close to 4,000, and then it ranges all the way to 8,000. The total number of RNA molecules is about 10 times that, and it's pretty evenly distributed, mostly on the lower end, but also, again, the little squishes, a few that are really high. And then our percent counts of mitochondrial, most of it is around 0%. Again, we want it to be very low, but we do have some high mitochondrial contamination as well, and those might be things that we would actually cut off later on, because this might be indicative of some cells that were just less happy or less healthy. So those are all saved in the figures folder here on the side here, but we can also see it here. We can also look at those metrics in a different way. We can do sort of correlation analysis. So as expected, we see that as we have more genes detected, we also have more RNA molecules. That makes sense. The more things we detect in a cell, the more diversity in it there would be. So you would see this type of curve. We can see also percent mitochondrial are all mostly down here, the ones with the high percentage. And actually they kind of offshoot a little bit. So this again is another hint that maybe these are worth removing or excluding. Next is doublets and ambient RNA cleanup. And so we won't run this part here because it is dependent on using R, or sometimes there's some Python tools as well, but it gets really complex. And so this is just something to consider as you're doing your own analysis. You might have to look into specifics and how to run these with the right tool for what you're trying to do. But the issue that we're addressing here is that when we do single cell RNA sequencing, We don't know if there's two cells that came out together, or if whatever came out with the cell was just floating RNA in the solution or RNA in the actual cell. And so there's tricks to find these. And SoupX, SC Double Finder, Scrublet, you can check out these links. They explain how they do this and what are kind of the tricks that they use to do this. But broadly, the idea is that, doublets is we simulate fake doublets from cell types that are in your data set, and then we find cells that look like those. Or with ambient RNA, we take that dplot and we take that empty space, the gray part at the bottom. We look at the RNA in there and we subtract it from the top part. And that's really simplified, so you might want to look into this a little bit more. So we won't run this right here. Okay, so next we're going to filter based on these QC metrics. So after we've done all these quality controls, we pick our metrics, again, driven by the biology. So some cells might actually normally have high mitochondrial content. Nuclei, for example, should have almost nothing because you've removed the nuclei. So these are all things to consider. And so what we're going to do here is filter cells that have fewer than 100 genes. We're also going to delete genes that are in fewer than 3 cells because they won't give us any useful information. We're also going to filter everything that has more than 5% mitochondrial RNA. And then we're just going to visualize this again just to see how it looks after that. So you can see it looks pretty similar. Both of these look pretty similar. Now we can see that we've stopped at 5% here, so we've removed a lot of that noise at the top, but really we didn't change anything here at the bottom. So we probably didn't remove a whole lot of cells or nuclei. And so it really won't affect the downstream analysis a whole lot, except just kind of make it a little bit cleaner. Again, all of this should be driven by biological knowledge. So if you understand your sample and your cell type, your cell source, then you should be able to make some pretty informed decisions about these cutoffs. So after you've done this, what you want to do is normalize because, again, we have different cells, we'll have different sequencing depth, just kind of from the luck of the draw or other aspects as well. And so what we do is we save the original data into another, what we call layer here, which we call counts. And then we're going to normalize that data. And then we're going to log transform it just to make it easier to work with and visualize. Yeah, and this step is pretty quick. Okay, so after that, what we do is feature selection. Now, we can try to do cell clustering and process the data by looking at all the genes. But the truth is that the genes that are going to be more informative about the differences between your cell types are going to be the ones that are most variable. And so to make this a little bit more efficient, we're going to use the top 2,000 variable genes. We tell it that there's a batch peak, just so we can do some batch correction if there is any batch effect. And then finally, we're going to do dimensionality reduction, because really we're talking about 5,000 cells or nuclei by 10 to 20,000 genes. That is just too many data points to look at. And so we need to simplify it somehow. And this is what dimensionality reduction does. And so you can look more into the depth and how these work. But two popular methods to do this is principal components analysis and uniform manifold approximation and projection, or UMAP. And the UMAP is usually what you'll see single cell RNA plotted as, or sometimes you'll also see t-SNE, which is another common way to do this as well. So what we're going to do here is actually run PCA. We're going to tell the code to find closest neighbors, or the cells that look more like each other. We're going to use that information for the UMAP. And then we're actually going to plot the UMAP, coloring it by the donor ID, and saving that image. I'm going to be called UMAP unclustered.png, but it will also come out here in the output as well. This could take a little bit of time, but it should complete here. Now we're going through the neighbors, right? So following that arrow, it's detecting the neighborhood graph, which will be used by the UMAP. Okay, so now we're running the UMAP. And now we're plotting that. And now you can see we've got some pretty clear clusters. Everything's called the same color because it's colored by donor. We haven't done clustering yet. But we could see some pretty obvious clusters here. So now we do clustering. So we want to find out what are the individual cell types. So first of all, we have to draw clusters of what cells are most like each other. And so to do this, we perform Leiden clustering, which is just a community detection algorithm, which is used to find, the most similar cell, the nuclei, based on this dimensionality reduction. So here we go. We run the Leiden clustering. We're going to choose a resolution of 0.1. This is something you'll have to optimize for your data set. We're going to do a couple of iterations, and then we plotted this. And now you can see we found 8 clusters. And they separate pretty much as you would expect. It's interesting that this one's split up into 2, but it might actually be 2 different cell types here that are just very similar as well. So it might be 2 subtypes, while these are, pretty different cell types. Okay, so how do we determine what those cell types are? How do we make sense of this? What we want to do is actually examine the data. And so there's several ways that we can examine that data. And so first, what we're going to do is actually just find the top 3 genes in each of these clusters that represent that cell type. So it's differential expression. We're looking for what is higher in this cluster versus everything else, for example, or higher in this cluster versus everything else. So this again should run relatively quick. We're doing every single cluster, and so we should have 8 different groups here. Sometimes you'll get warnings. Usually it's not a huge problem, but it's just something to pay attention to. Okay, so this is the plot that we have here. It's a dot plot, and it represents the top 3 markers again, right? So you can see that in each of these clusters, you have 3 dots. And the size of the dot represents what % of the cells expressed that gene, and the intensity of the color reflects the mean expression. So we have things that are expressed a lot, but low. We have things that are expressed a lot and really high. And so you can see that in this cluster 6, we have a lot of HBV genes, so I believe hemoglobin genes. Cluster 6 is this pinkish one, so it's this tiny little cluster here. Maybe some blood cell contaminations. Here, 0, we see things like ASS1, which I know is a hepatocyte gene. Blue, which is another hepatocyte gene, but there are 2 different zones of the liver. SLC8A1 is a macrophage marker. And these are things that I know from working with liver. So you'll know your data set and your sample a little bit more. You'll have to look through literature or databases to find some information on these. STAT2 for endothelial cells. And I don't know exactly what the other ones here are, but using this data, you can already kind of tell what these cell types are. Another way you can do this as well is look specifically for markers. So, for example, I mentioned ASS1, which is pericentral hepatocytes. Central hepatocytes are blue, and macrophage are SLC8A1, and endothelial cells are STAT2. We can look at those specific on the UMAP. And you can see, so these are probably portal hepatocytes, central hepatocytes here, macrophage or Kupffer cells here, and endothelial cells here. So again, we can see that we're starting to see what cell is which. And so this is how we start annotating these cell types. It's a combination of finding the most similar cell types, looking at the enriched markers, the marker genes, understanding the biology, which is critical, understanding the literature and knowing what those markers are, and sometimes discovering new markers that we didn't expect. And these are things that we can sort of follow up on when we identify them. Maybe they co-localize with other markers. But this is where we can really kind of have fun with single cell data and really kind of learn a lot. So this is where I'm going to leave you for this section, and I'm going to come back to you with another video with just a quick wrap up, and hopefully this will have been helpful to you. Thank you.
We have one question.
Yeah, sure.
I was curious, in regard to doublet discrimination, if I have samples that are polyploid either due to endoreduplication or somoclonal variation, would those be discriminated against in that, or would I be able to use just a standard model?
So those shouldn't be as much of an issue. When it comes to doublets, these doublet detection tools do really well when it's mixed cell types. Because you are simulating sort of combinations of both and trying to delete those. And so finding like doublets of the same cell type are actually really hard because they just look like there's more of the same genes, right? It just looks like it's more of that same cell. So yeah, if you got like polyploidy or anything like that, then it shouldn't really be something that you lose. But that is something where, I mentioned a lot talking about, you want to drive your analysis by the biology. You don't have to do doublet removal. Sometimes the rates aren't that high. The rates are higher the more cells you load. And so it might be something of maybe you don't load as many cells just to be extra careful or something like that. But that's where like knowing your biology, the biology you're exploring, can really help making sure you're picking the right tools and approaches for the analysis.
Okay. Does anyone ever include the live imaging of cell sorting when they're doing single cell sequencing experiments ever? Your knowledge?
Oh, I'm sure someone has. That technology for sorting, I think, is pretty new and it's pretty impressive. So I haven't seen one. Well, I shouldn't say that. The plate-based methods, people would image those. If you did on a 384 well or a 96 well, there are some examples, but I haven't seen any like published pictures or like correlation to images of cells or anything like that.
Okay. I wasn't sure if that would help maybe with the polyploid sort of proof of doublet discrimination.
For doublets, definitely.
Yeah. Okay, great. Thank you so much.
Yep.
There's just one more question. Oh, maybe not one more. But a really quick question is, towards the end of the analysis, you showed us the different biomarkers or maybe markers of specific clusters within your UMAP. So I'm wondering if we know, for example, we have like 3 or 4 genes of interest, and we only want to enrich for those cells which are expressing for those or differentially expressing for those genes of interest, and only take that subcluster in our analysis instead of all the cells. Is there a way to do that?
Yeah, so there's 2 ways you could do that, right? One is you could do it experimentally. So you could use like you could sort for those cells. So I've seen several papers, they'll sort different types of cells separately, and they'll only sequence or do single cell on one half of them. But you can also go into that data and say, give me all the cell type X, and then pull those out. And then I want to explore those more. So one thing we did that with is for liver, we took just the hepatocytes and looked at the spatial pattern of the hepatocytes. Because you can really see those patterns in the hepatocytes, but not so much in the other cell types. So you can definitely, after you figured out what cell type you want to look at more closely, you can take those out and then analyze those separately as well.
Oh, sorry. Was there another question? Okay.
Okay, so we also have one ticket to the McDonald Genome Institute tour on Wednesday to raffle off to the people that are here. But is there anyone who does already have a ticket to it? Or like to a tour on Wednesday? Because we'll just enter the people that don't have a ticket to a tour on Wednesday. It's the animal side one at the McDonald Genome Institute in Washington University on Wednesday from like 12:45 to 5 p.m.
Start off with the Millipore Sigma facility, and then we'll
Yep.
cover the McDonald Genome Institute.
Yeah.
We're just. Okay, thank you, Rance.
All right, thank you. I just want to mention, if you're ever running through this code, I mean, it's available to you even after this workshop. Feel free to contact me. I'm happy to answer any questions, and if help with the going through this notebook. Yeah, feel free to contact me. Thank you.
Thank you.